Today, I'm going to talk to you about GcMAF, which is also known as Globulin-component Macrophage-activating factor and was first discovered in 1991 by Japanese immunologist Dr. Nobuto Yamamoto. So what exactly is GcMAF? Well, GcMAF is a protein derived from the modification of the vitamin D binding protein, which is part of the vitamin D pathway that transports vitamin D metabolites between the skin, liver and kidneys. There is around 1/1000th of a gram found within healthy individuals, and the immune system does not function adequately without it. GcMAF signals the activation of macrophages to a tumocidal state, therefore it plays a vital role in immunity within the body. Without the presence of GcMAF, our bodies are in an immunosuppressed state, as macrophages are not able to successfully engulf cancerous cells, and we lose a further 19 known attacks the body has on cancers. So now we're going to take a look at how GcMAF is made naturally within the body. GcMAF starts life as a vitamin D binding protein, which is synthesized within the liver by hepatocyte cells and is modified by the immune cells to create GcMAF. When the vitamin D binding protein comes into contact with the enzyme beta-galactosidase, which is embedded in the outer cell membrane of B lymphocytes, it removes the sugar D-galactose, which in turn activates the B cell. The molecule will then come into contact with T cells, which are also known as T lymphocytes. Located in the outer membrane of T cells, there is an enzyme called solididase, which removes the monosaccharide salicylic acid from the molecule, which forms the structure GcMAF. This in turn also activates the T cell. So as GcMAF is a natural human protein, what exactly happens to the production of this protein within the body? Well, first we need to take a look at an enzyme called nagalase, which is also referred to as alpha-N-acetyl-galactosaminidase. So what exactly is nagalase? Well, cancer and some virus-infected cells such as HIV and influenza secrete nagalase, which is an extracellular matrix-degrading enzyme. Nagalase testing can give early indications of cancer in direct comparisons to cancer's abundance and tumour size within the body. Nagalase completely deglycosylates the vitamin D binding protein, which is the precursor for the macrophage-activating factor. When the vitamin D binding protein is deglycosylated, it loses its cell-mediated response mechanisms on macrophages and has no macrophage-activating ability, which leads to immunosuppression. As nagalase is a catalyst, one molecule can completely cleave all the immune-activating glycoproteins off the vitamin D binding protein many times over, as it is not consumed in this reaction. So now we're going to take a look at how GcMAF is made for therapeutic applications, or extracted. So GcMAF is extracted from healthy blood serum or milk, and using a 24-step method which includes deglosylation of the vitamin D binding protein, it is then placed in vitro and suspended in a saline solution due to its mass. GcMAF can be made via recombinant DNA technology. This is achieved by inserting a gene that codes for the vitamin D binding protein into the bacterium E. coli. This piece of DNA reprograms the bacterium to secrete the vitamin D binding protein, which is then submitted to a 24-step process that includes deglosylation, the same process it undergoes in the body. This now leaves us with GcMAF, which is the GC protein with only a single sugar attached. This molecule now has the ability to go around the body and activate macrophages to a tumocidal state. Before GcMAF is administered, it undergoes a purification process. This ensures the sterility of the product. GcMAF is either administered via intramuscular injections or nebulization. Now let's take a look at what happens when GcMAF is administered into an immunosuppressed patient. Well, GcMAF activates macrophages. This allows phagocytosis to destruct cancer cells and bacteria. Macrophages activated by GcMAF have shown a considerable variation of receptors that recognise the abnormality in malignant cell surface and are highly tumocidal. GcMAF has also shown to inhibit angiogenesis. This means it stops the blood supplies to tumours, and this starves the malignant tissue from the nutrition needed to grow and survive. GcMAF has shown to increase the rate of apoptosis, which is the programmed cell death of cancer cells. It also reverts the cancer cell's phenotype to normal. This means it turns cancer cells into healthy cells. Research also shows it reduces the metastatic potential of human cancer cells in culture and increases energy production at the mitochondrial level. When GcMAF enters the body, nagalase does not recognise the molecule, as it only has the one sugar attached, and nagalase can only completely destroy its precursor, the vitamin D binding protein. So things to note. Well, GcMAF has little or no side effects, apart from the potential reactivation of latent viruses. However, the rebuilt immune system may in some cases cause minor secondary side effects, but this is more common with late stages of cancer. It can also cause side effects with people with autism and HIV. This is because the viruses fight back against the rebuilt immune system's attack. This is a completely natural treatment, therefore completely ethical, as all of us healthy individuals have it inside us naturally. GcMAF has limited effects on patients that have received radiation and chemotherapy, as these treatments kill the white blood cells that house our immune system. It has positive effects psychologically by pain relief and the potential of remission from cancer if the treatment plan is followed correctly. Trials conducted by a company called First Immune Biotech in Guernsey have shown up to a 50% tumour reduction in just 2 weeks after the initial administration. So now we're going to take a look at one of the traditional methods for treating cancer, chemotherapy. Doxorubicin is a chemotherapy treatment sold under various trade names. This is a treatment for many different types of cancer. This drug has shown to slow or halt the growth of cancer cells by inhibiting the enzyme called topoisomerase II, which cancer cells need to divide and grow. This is either achieved by apoptosis or meiotic catastrophe. However, this drug does affect the cellular mechanisms within healthy cells. The drug usually is directly administered into the bloodstream through injection, although there are other methods of administration that can be used. There is no pill form of this drug, as it is viscous, which means it causes blistering on the skin. Doxorubicin is often used together with other chemotherapy drugs. This is because the different drugs have different mechanisms of action within the body. This prohibits cancer building up a resistance to chemotherapy. So now we're going to take a look at comparing GcMAF to doxorubicin. So first we're going to take a look at the side effects. Well, as discussed previously, GcMAF has minimal or no side effects. This is because it's a natural human protein. However, side effects can be apparent in the later stages of cancer. It has also been observed that those with autoimmune diseases can react severely to the activated immune state, so research into the management of autoimmune reactions while taking GcMAF is important. No deaths have been recorded from a direct result of GcMAF. The side effects observed from doxorubicin can range from fatigue, sickness and vomiting, to in some extreme cases even death. A study conducted by Public Health England and Cancer Research UK found that across England around 8.4% of the patients with lung cancer and 2.4% of the patients with breast cancer died within a month of the treatment. Hair loss is also a common side effect from chemotherapy, which can cause the obvious psychological problems. As chemotherapy also kills white and red blood cells, susceptibility to infections can increase. This is along with lowered oxygen levels within the blood. The list of chemotherapy side effects are extensive. This may be due to the number of patients that have undergone this treatment in comparison to GcMAF. GcMAF's indirect effectiveness on cancer is very good, as it restores the immune function, which then allows macrophages, NK cells and other cells to kill the malignant tissue. The effectiveness will depend on many things. However, the most important thing is adequate vitamin D3 levels and a good diet full of lipids with minimal sugar intake. GcMAF has also shown to lower nagalase levels, which restores the body's own natural production of GcMAF. The effectiveness of doxorubicin on cancers can vary, as with all available treatments. Chemotherapy can treat cancer cells almost everywhere in the body and is more effective and safe to use on the earlier stages of cancer. However, new studies have demonstrated that when secreted, it can interact with nearby tumour cells and cause them to grow, invade, and importantly resist subsequent therapy. According to the Fred Hutchinson Cancer Research Center in Seattle, Washington, and their paper on protein WNT16B, cancer cells become resistant to chemotherapy, which makes the disease worse. So what about the origin of these treatments? Well, as GcMAF is a human protein, it's a type of natural therapy. This coincides with today's blossoming demand for natural sourcing for medical treatments and is therefore, from an ethical standpoint, a strong contending treatment for cancer. So what about the origin of doxorubicin? Well, first we need to take a look at daunorubicin, which was first isolated in the 1950s by an Italian research company called Pharmatalia. Research began an organised effort to find anti-cancer compounds from soil-based microbes. A new strain of Streptomyces persitus found in a soil sample produced a red pigment. This was then isolated, and an antibiotic from this bacterium was effective against tumours in mice. Clinical trials began in the 1960s, and it was found to be a successful treatment of certain cancers. However, by 1969, it was recognised that daunorubicin could lead to fatal cardiac toxicity. Researchers at Pharmatalia later discovered that changes in the biological activity could be made via minor changes in the compound structure. A strain of Streptomyces was mutated using a highly reliable carcinogen, and this new strain produced a different red-coloured antibiotic. They named this new compound doxorubicin. So what about the NHS availability for these treatments? Well, unfortunately, GcMAF and most other types of immunotherapies are not available in the NHS. This is where cancer treatments become political, and this may be due to the 1939 Cancer Act. However, most types of chemotherapies are available in the NHS, but the single cost for around a chemotherapy, if gone private, can be in excess of £2,000. Now let's take a look at the chances of secondaries after receiving one of these treatments. Well, if GcMAF is successful, the chances from secondaries from the same source is very rare. This is because the GcMAF would have restored the immune function to act on cancer cells as it does all of our lives. The chances of secondaries for chemotherapy are likely, as the root cause is not being treated but only the malignant tissue that's present. This can allow cancer cells to dissociate with the malignant tissue and re-enter the bloodstream to mutate once again. So lastly, let's take a look at the tumour destruction rate. As discussed, GcMAF can halve the sizes of the tumour within 2 weeks of initial treatment, and diet has shown to be a major contributing factor to this. This is the fastest destruction rate in comparison to any type of chemotherapy. The exact tumour reduction rate doxorubicin has on malignant tissue is uncertain, as it is usually combined with other chemotherapy drugs. I am now going to show you a short video that shows activated immune cells undergoing some of the cellular processes discussed in this video. That concludes my video on GcMAF. Thank you for listening, and I hope it did make some logical sense. If you do have any questions, please do not hesitate to ask.